Antibiotics derive from microorganisms that compete with each other in the natural world. A Danish research group has now developed a battlefield on which bad pathogens meet good antibiotic-producing bacteria. After the battle is over, the researchers examine the bacteria's chemical weapons to see whether these may also help people defeat the bad pathogens.
So we use this competition between the good and the bad bacteria to filter out all these potential biosynthesis products, all the potential antibiotics which aren't active against the bacteria we actually want to treat. Starting from these thousands of bacteria that we study initially, we will reduce them to a much smaller number.
The researchers have named this bacterial battlefield competition-based adaptive laboratory evolution because bacteria accelerate through an evolutionary process. Robots speed up this process.
You can of course do this manually, but this is a lot of handwork and very slow. So we have developed a robotic system that can do this co-cultivation. So the robot can put the antibiotics producers on agar plates, can overlay them with pathogens, can pick them to new plates. So we can do these, what we call competition-based adaptive laboratory evolution experiments in high throughput with many strains in parallel.
Instead of testing one antibiotic at a time, the researchers can thus test billions of potential antibiotics simultaneously, even without knowing which ones are being tested.
Under these conditions, these bacteria switch on the production of this antibiotic, which then, in the end, leads to the formation of inhibition zones. In the beginning, maybe very small, but after we have continued our procedure, which takes several weeks, these inhibition zones get larger and larger. And these inhibition zones are caused by small chemical molecules that inhibit the growth of the pathogenic bacteria, and very often they even kill them.
When the battle is over, the researchers scan the DNA of the good bacteria using computer programs to determine which genes are responsible for producing the chemical weapons. This is called genome mining.
Remove them from the original producer and bring them into some bacteria which we know much better, and use these bacteria as some kind of cell factories. So where we want to produce the antibiotics from our original soil bacterium in bacteria where we have a lot of protocols available, we know how to grow them, what they like, what they don't like.
The goal for the next 6 years is to identify and produce at least 50 candidates for new antibiotics to combat the resistant bacteria, which present a massive challenge to health systems worldwide.
We can use it for many different pathogenic bacteria. That's why we like it so much, because it gives us a huge opportunity to find new molecules that are active against the most dangerous pathogens that are currently out there.